So good afternoon. Thank you all for being here in person and over Zoom. We have the mental health and addiction matrix and monthly rounds today. And it's my pleasure to introduce Dr. Anil Maharaj, who is an assistant professor in the UBC Faculty of Pharmaceutical Sciences. He's a pharmacist and a researcher doing clinical pharmacokinetics and pharmacometric studies. And he will be joined later by Sahithi Thotakura, who is his PhD student, also in the Faculty of Pharmaceutical Sciences. Sahithi has a PharmD in pharmacy, and she was a former junior scholar with the Matrix N cluster. So we're very happy to welcome them both today, and thank you for joining us.
Great, thanks for inviting us, Lily. So I'm Anil Maharaj. I'll be talking about some of my work looking at pharmacometric modeling in the space of opioid pharmacology. So what is pharmacometrics? We'll talk about that in a second. Okay, let's see here. Well, here it is. So pharmacometrics is essentially the field of research that seeks to describe the interaction between drugs, patients, and it does this by interlocking biology, physiology, and pharmacology through these mathematical models. So in terms of what do these mathematical models look like, you can see that they can be something very simple where you visualize the body as just a single compartment, or it can get a little bit more complex where you have two compartments representing the body, or you can ratchet up the complexity completely where it looks like you have a compartmental model on steroids. Here you have a compartment for every single organ system in the body, and these are known as physiologically based pharmacokinetic models. So depending on what you're trying to do with your model, it will kind of dictate what type of model you will need to develop in your research. So no matter how complex your models get, in essence, what these pharmacometric models are aiming to do is to describe something known as the dose-exposure-response relationship. So theoretically, there's this innate relationship between taking a dose and an internal exposure in the body, and then that internal exposure precipitating or mediating some type of effect. So you can think of this as you take a dose of a drug, you get some specific plasma concentrations, and it affects some mediator in your body. If you're thinking about opioids, you take some opioids, get plasma concentrations in the body, it gets to its target site, and you may see analgesia, but you may see the adverse effect of respiratory rate depression. So by understanding or providing a quantitative framework to describe that dose concentration effect relationship, pharmacometric models can serve in a variety of roles in terms of the drug development process. The first will be lead compound selection, the second, optimizing trial design, and the third is human dose optimization. So if we think about lead compound selection, this is basically asking the question, which compound in these early preclinical stages, when you have a whole ream of compounds that can potentially go into the clinical sphere, which one of these are going to have the best success for exceeding clinically? It can also, once you've selected that compound, it can help you optimize your trial design. So you have this compound that you're moving forward in your clinical or preclinical studies, and you want to figure out sort of, how do I prove it's working? So by having that mechanistic understanding of the relationship between dose, concentration, and effect, you can basically use it to inform your designs of your studies, either figuring out how to best sample either the concentrations or effects in your study design, or even how many subjects that you need to prove statistical significance that your drug is having the desired effect. And last is human dose optimization. So once you get into the clinical sphere, the question is, what dose of my drug do I need to effectively treat whatever I'm looking to treat? So this idea of dose is very important. And as we know, dose is kind of this conundrum. And it was really exemplified by Paracelsus. So this is a German-Swiss physician back in the 1500s, known as the father of toxicology. And he basically said this statement that was very simple but profound. So what is there that is not poison? All things are poison and nothing is without poison. Solely the dose determines that a thing is not a poison. So it really hits at this heart of this idea that these drugs that we use to treat all sorts of therapeutic or pathophysiological processes in our body, these very same drugs with a therapeutic effect can also elicit a toxic effect. And the idea of what mediates a therapeutic or toxic effect is really part and parcel of the dose we give patients. So the idea here, or where pharmacometrics really comes into play, is it can help researchers identify the doses that increase efficacy while minimizing toxicity. So we'll talk about 3 different examples of pharmacometric models in the space of opioid pharmacology and how it can be used to identify dosing. So in terms of what types of pharmacometric models we use in the drug development sphere, they can range in terms of 2 classes, either empiric or mechanistic models. So when we think about empiric models, these fall under a class known as population pharmacokinetic or pharmacodynamic models. And the idea here is that these are very simple models, and the structure isn't related to any particular aspect of human physiology or anatomy. And the parameters that we get for these models are determined in a top-down or data-driven fashion. So the idea of how you would fit these models looks something like this. So say you start with a clinical study. You give a drug to a set of patients. After that drug is given, you either measure concentrations or effects, and you get data that maybe looks something like this. You have plasma concentrations first time after giving a particular drug. What you would do is you would take that data and you would analyze it using one of these population pharmacokinetic or empiric models. And the whole idea of the structure of these models is fit under the guise of something known as parsimony. You want the simplest model to describe your data as possible. So this data is being used or is being described by that pharmacokinetic model. And by describing your data or the pharmacokinetics, this dose concentration relationship, you can use this model to drive a whole bunch of hypotheses. So using my model, now that I understand the relationship between dose and concentration, how can I figure out what dose I would need to give patients of different sizes? So for example, here I've taken this model and I've used it to run a dose simulation for subjects of different ages. So if this was a pediatric study that included a bunch of patients from 2 to 18, I would be able to use that model to help derive dosing in that same population. So the advantages of this approach is it's quite simple and it's flexible for describing the data. You can throw into this model all sorts of unbalanced study designs where you'd have 1 or 2 samples per patient, but still get out rational estimates of the dose response or the dose concentration relationship. One of the disadvantages is it really lacks the capacity to extrapolate. So the population that you studied, so say it was children, that's the population you're going to run your dose simulations in. You really can't get outside of that unless you make some overarching assumptions based on your model. But for better or worse, it really can't extrapolate outside what you've developed the model on. The next type of models are known as mechanistic models. And these fall under those models that I was saying look like a compartmental model on steroids. So these are known as physiologically based pharmacokinetic models. And you'll notice that they have a physiologically relevant structure. Instead of the parameters being fit based on the data, these parameters are going to be fit bottom down or in a knowledge-driven approach. So the idea here is you take your understanding of the drug you're working with and your understanding of organism physiology and anatomy and some idea of your drug of how it's absorbed, distributed, metabolized, and excreted, this anatomy data. And you throw that together into something known as a physiologically based pharmacokinetic model to end up something like this. So you'll notice physiologically relevant structure. You have a whole bunch of compartments representing different organ spaces in the body. And the whole idea is to make a virtual human. And Sahithi will be talking to you a little bit more of these type of models in her research. So once you have this model, you can use this to help you develop dosing simulations, very similar to what we had before. But the idea here is it didn't really require any patient data. We just used our fundamental understanding of drug physicochemistry and organism anatomy and physiology to derive or estimate the pharmacokinetics in these populations. So the advantages of this approach, it really comes from that extrapolation. So for example, we're kind of segregating our parameters based on drug physicochemistry and organism anatomy and physiology. So if we really want to predict the pharmacokinetics or the disposition of the drug in a different population, all we'd have to do is just change these parameters and run a new simulation. So using this model based on these bottom-up fundamental concepts, how drugs interact with the body, we can use this model to extrapolate outside of the populations that we develop these models in. The big disadvantage of this, as you can see, it's quite complex. And the other piece is, in terms of a regulatory standpoint, when we generate predictions, they usually require some type of clinical confirmation. Because as you can see, up to this point, there's no really clinical data informing the model development for most cases. Okay, so now I want to briefly chat about 3 different examples where we've incorporated pharmacometric modeling in the space of informing opioid dosing. And the 3 examples I'm going to talk about is looking at fentanyl dosing in children with obesity, looking at remifentanil dosing in adult patients with obstructive sleep apnea, and the last one is looking at buprenorphine in adults with opioid use disorder. So let's start with the fentanyl. So this idea of fentanyl and pediatric obesity. When we look at sort of the prevalence of obesity across Canada and America, we notice Canada, we're doing a little bit better. This is 2018 estimates, but it's reported that 10% of children in Canada have obesity. It's double that in America, 20%, but if you see the size of the meals that you get there, you would really understand that. In terms of how do we define obesity in children, it's a little bit different than adults. So in adults, we just look at the BMI, right? And we say, whatever your BMI is, we have an absolute value, which is the demarcating value that we say is obese. So greater than 30 BMI, we consider an adult to be obese. But since children kind of develop and grow and have all different sizes, instead of this kind of one line marker of a BMI, we instead use the 95th percentile of a child's BMI for age and sex. So the BMI that a child is actually determined to be obese will change based on age and sex up to 18, and then they fall back on this 30 BMI to classify as obese. And the idea is that the impacts of pediatric obesity on drug pharmacokinetics, so this idea of the dose concentration relationship and drug dosing, are relatively unknown. So when a child gets larger, of course, they have more adipose tissue. And this can change the way drugs distributes in the body. But not only that, a drug can be metabolized in a different way in an obese patient versus a non-obese patient. So we notice that in obese children, we see increases of blood flow to specific organs in terms of in conjunction with an increased body weight. There have been reports of increased glomerular filtration rates. So the idea here is these children might clear renal drugs or drugs that are renally cleared quicker than their non-obese counterparts. And as well as the enzymes that mediate metabolism or the major metabolism of the body, either phase 1 metabolism like the CYP enzymes or the UGTs like the phase 2 enzymes may change with obesity. Some may increase, others decrease, others stay the same. So obesity not only changes the way the drug distributes, but potentially how the drug is eliminated from our body. All this goes to say is how you dose an obese patient may differ than how you dose their non-obese counterparts. So the idea here is this project came out of collaboration with the Pediatric Trials Network, and this is a conglomerate of pediatric research institutions, mostly in the United States, that has a concerted effort to study pediatrics, obviously by the name. So the idea here is that children, it's difficult to study. Recruitment in these clinical trials is quite difficult. You're thinking you're already dealing with children who are sick, which is a very small population number. Recruiting them is very difficult. There's logistic difficulties in terms of consent to assent. There's also difficulties in studying them. Children, think a newborn child has a blood volume of the size of a can of Coke. How are you going to conduct a pharmacokinetic study if that's all the volume you have and you need so many samples? So fentanyl is a common opioid or high potency opioid that is used not just as a drug of abuse, it's also used in critical care settings as well as surgical settings. So the idea with this Pediatric Trials Network project is that for these children who are admitted to hospital undergoing some type of procedure, what type of dose of IV fentanyl would you give a child with obesity? As this is when I was working in the States, 1 out of 5 children at the time were considered to be obese. So the role of pharmacometrics in this project was to characterize the dose concentration relationship in fentanyl in obese children who underwent something known as opportunistic sampling after receiving sort of their standard care dosing of IV fentanyl. So the idea of this Pediatric Trials Network, by going to so many institutions, you can actually help sort of that low sample size that you would have been experienced if you only dealt with an N of 1 or an N of 3 institutions. By going over 80 different institutions, you begin to collect information from a larger population of pediatric subjects. So this idea of opportunistic sampling, it really capitalizes on this idea that children are logistically difficult to study. So opportunistic sampling is the approach where when a patient gets something like a typical laboratory or light straw, we will take the excess sample and quantify the drug or fentanyl in that concentration. So it really costs, in terms of poking the patient, nothing to conduct these opportunistic sample designs. And they're quite intuitive and used commonly in pediatrics to deal with this low recruitment or low sample size. So the idea of the pharmacometrics is to provide a dosing recommendation for children with obesity. Yeah.
We've got lots of talks here about fentanyl and other opioids. Can you just give us a sense or two as to why they feel that fentanyl is a good prospect to deal with childhood obesity?
Not to deal with childhood obesity, but just when they're being treated for surgery or when they're being treated procedurally. Yeah.
So the purpose of giving the child that is not to deal with their obesity.
No, that would not work whatsoever. Okay. So just taking a look at this data, this is the data from the study. You'll notice how quite sparse it is. This is the concentration time data we got. And we were only able to collect out of over 80 institutions data on 32 children and 53 plasma samples. That means that some children only provided at most one sample, others provided 2 samples. So we have very sparse data from children from 2 to 19 years of age, from 16 to 165 kilograms. And we're trying to get that data and get something rational out of it to help us understand how does the dose response or the dose concentration relationship change in these patients with obesity based on very sparse data. And this is where this population or empiric modeling really helps us out. Based on statistical modeling approach and these data fitting algorithms, it is able to extract a maximum amount of information from these unbalanced study designs where you only have 1 or 2 samples per patient. So from that data, we created this population pharmacokinetic model. And what we did with it is we used this model to help us drive these hypotheses or testing of doses that are recommended for these patients. So using this population pharmacokinetic model, the first thing we did is we took a look at what are the label recommended doses for children receiving IV fentanyl. And we tested them to see how well do they align with a theoretical sort of therapeutic index between 1 and 3 nanograms per ml. So giving these patients 1 microgram per kilogram per hour, you notice the green line, the probability of them landing within this therapeutic index is around 60%, and it's relatively flat. But you'll notice that red line, the probability of them exceeding that threshold is quite a bit high and increases as their weight increases. So this idea of giving a weight normalized dose to these patients, it's really doing them a disservice in terms of the prevalence or the probability of toxicity. So after testing what is given to patients, we also use the model to say, what would be an ideal way to dose these patients? And it really came down to a simple formula that you divide by the weight to the power of 0.25, and it'll give you a decreasing weight-based dose as the patient increases. But you'll notice with the model simulations, what it's indicating is that the probability of a therapeutic and the probability of a toxic response remains consistent throughout the weight range. So a very simple equation derived from this model based on sparse data, and it allowed us to come up with something profound. How do you dose these patients? So again, not treating the obesity, treating whatever pathophysiological that pathophysiological disease state that they have during a surgery or procedure. So, yeah, pardon me? Yes, analgesic, yeah. So the innovation of pharmacometrics in this case is the analysis of sparse data from multiple children with obesity to come out with something rational. Okay, let's take a look at another example where we have applied pharmacometrics to adults, but this time with obstructive sleep apnea. In this case, very similar. The remifentanil is not being used to treat obstructive sleep apnea, but I promise the last case, there will be an opioid that's using to treat something. So obstructive sleep apnea, it's this reoccurring airway collapse during sedation or when a patient is sleeping. So the idea here is it is quite prevalent in adults. In Canada, it is almost estimated that 1 in 3 adult patients or adults would have sleep apnea or undiagnosed sleep apnea. Now, patients with obstructive sleep apnea are inferred to be more sensitive to the effects of opioids with an increased risk of respiratory depression with that airway collapse that is happening nocturnally. So if you look at clinical guidance, it reports that there's an increased effects of opioids in OSA But when looking at it, it's really, if you take a look at the guidelines, it's based on what falls under that low levels of evidence, either small uncontrolled studies or expert opinion. So the question here is, should opioid dosing be adjusted in patients with OSA? And how can we use pharmacometrics to help us answer that question? So here, we're going to use the pharmacometric modeling approach to help us understand not just the dose concentration, but the dose concentration effect relationship of remifentanil in adult patients with either treated OSA, with either CPAP or BiPAP, untreated OSA, or patients with OSA and treated with one of these CPAP and BiPAPs. So by using these three different patients, we have, does OSA have an effect? Or does treatment of OSA with CPAP or BiPAP have an effect? And the idea here is we want to use this to provide guidance in patients with obstructive sleep apnea with or without treatment. So what we did, instead of sparse sampling in adults, there's a larger blood volumes. Logistically, we don't have to worry about this idea that you can't get consent directly from the patient. All I'm saying, it's a great place to be conducting these type of studies because you can take multiple samples a lot of the time, and it's more ethically palatable. So you'll notice that we have a rich concentration dose response data set. We had 73 adults, and each adult we collected more than 10 plasma concentration samples to define the pharmacokinetics of remifentanil. So you can see here is that each patient got increasing doses of remifentanil, and after each sort of increasing doses, we took the concentration. After about 4 dose increases, we shut off the infusion, and it decreased the concentrations. So while we were increasing the doses of remifentanil, or the concentrations of remifentanil, we also measured various effect measures. Here we measured pupil size. So pupil constriction is or meiosis is one of the most sensitive measures to opioid effect. We looked at their thermal analgesia. So would the patients not react to a thermal diode placed on their skin, as well as their end expiry CO2 looking for respiratory depression. So by combining all these data across doses, concentrations, and effects, we were able to create a very similar pharmacokinetic model. But this time, we added an extra compartment that equilibrated with that central compartment to now model the effect. So we get the dose concentration relationship, but now the concentration effect relationship. So what did we find from this pharmacometric models? The first thing is this is looking at the dose concentration relationship between 3 different groups. So without OSA, people with OSA but weren't treated. And then the last one is people with OSA and treated with CPAP, BiPAP. Basically, they don't look very much different from each other. So there's no difference in OSA in terms of the pharmacokinetics or the dose response relationship. How about the effect relationship? Are people with OSA more sensitive to the effects of opioids? We also found that the concentration effect relationship between the 3 different groups were not significantly different based on our models testing or hypothesis testing. So again, no difference in OSA. So it's very interesting that this model-informed sort of dosing recommendations or assessment really challenged that conventional notion that OSA alters the sensitivity of opioids in adults, basically saying that dosing adjustments might not be needed based on the dose concentration or the concentration effect relationship. The big caveat of this research is all these patients were still awake. We weren't able to give them remifentanil doses to induce sedation. So I think that is kind of the one missing, fly ointment, a missing piece of the puzzle. So the innovations of pharmacometrics in this case is it is able to analyze this rich data set across multiple different patients, but also multiple different measures, either concentrations, effect measures, and expiratory CO2 to come out with a rational understanding of the sensitivity of patients with OSA to opioids. Okay, the last example is optimizing buprenorphine dosing in patients with opioid use disorder. So this picture, downtown east side of Vancouver. So I'm pretty sure all of us have seen what it looks like down there. And it's very different than any part in Canada that I've been. When you look at sort of the impact of the opioid epidemic, especially in Vancouver, you'll note that we are the hardest hit across the whole country. On a nation level, Canada's third highest prevalence of opioid deaths. And when you look between 2016 to 2022, 34,000 Canadians have lost their lives, and this highest number of opioid-related deaths are in BC, with Vancouver being the epicenter. So what is fueling these unregulated opioid deaths? A lot of times, or what we're thinking, or the inference here, I don't think it's an inference, I think that's well out of the gate of what is causing it. It is fentanyl, or the presence of fentanyl in the illicit drug supply, or unregulated drug supply. So you'll notice that the rates of fentanyl deaths have increased over the last decade. And in terms of the prevalence of opioid in these, or the prevalence of fentanyl in these unregulated opioid deaths, what you'll notice is that the prevalence of the fentanyl has been increasing as well in these unregulated deaths. Almost that you can superimpose the presence of fentanyl in those unregulated deaths with the rates of opioid deaths. So this is kind of just proving that fentanyl is really fueling this opioid epidemic. So when we talk about the primary goal for treating patients with opioid use disorder, the whole idea is cessation of unregulated opioids. That's easier said than done, right? When we're talking about people using unregulated opioids, there is a psychosocial addiction as well as physical dependence. It's not like we can just tell them to stop cold turkey. This will result in very serious withdrawal symptoms. We're talking tachycardia, abdominal cramps, severe diarrhea, tachycardia, but life-threatening arrhythmias. So stopping cold turkey is not something that these people in our community would be apt for doing, and it's not something as a healthcare provider that is really recommended considering the severity of withdrawal symptoms. So what do? Essentially, we put them on something, or the recommended treatment is opioid agonist therapy. And this is just a fancy way of saying, let's get them on another prescription-grade opioid. So the two workhorses of opioid agonist therapy to convert someone off unregulated opioids are buprenorphine and methadone here in North America. So I'm going to talk a little bit about buprenorphine. So buprenorphine is an orally available, high-affinity, partial mu opioid receptor agonist, and it's indicated for treatment of opioid use disorder. This idea of a partial agonist comes from the sort of the pharmacodynamics of it, that it binds to the mu opioid receptor, but it doesn't elicit a full effect. So you'll notice a full agonist, it binds to the receptor and you get a full or 100% effect, whereas a partial agonist will bind to the receptor, fully bind to the receptor, but it will never fully activate the receptor. Therefore, you do not see a full effect. So this really comes in an advantage that when you look at buprenorphine, it kind of has this ceiling effect in terms of its opioid activity. So as you increase your buprenorphine dosing, you begin to see a ceiling effect when it comes to the intensity of opioid effects like respiratory depression. So you keep increasing buprenorphine, but eventually you will see its effect on respiratory rate plateau. Compare that to something like a full opioid agonist, like fentanyl, morphine, oxycodone, you're going to see continuing increases in respiratory depression with dosing. So that it's perceived that there is some safety built in to using buprenorphine. So, I mean, this sounds all great. What are the issues with buprenorphine? The biggest issue converting someone using unregulated opioids onto buprenorphine is this idea of precipitated withdrawal. So buprenorphine has high affinity for the mu opioid receptors. It binds very quickly, and it stays there linked to that mu opioid receptor for longer than something like fentanyl. So when it binds, it actually can displace fentanyl that was bound to the mu opioid receptor. And what this leads to is a patient who is on a full opioid agonist. Now their receptors are being bound by a partial mu opioid agonist, and it's not fully innervating the receptor, so they're not getting a full opioid effect. So they're at an opioid deficit. And what this can really lead to is these patients in these early stages of starting buprenorphine therapy is something known as precipitated withdrawal. Basically, you're giving them this opioid use therapy or opioid antagonist therapy, and you're causing withdrawal. So you'll see things like nausea, vomiting, sweating, diarrhea, abdominal cramps, tachycardia. So this has really prevented sort of widespread use or implementation of buprenorphine in these populations with opioid use disorder, leading to poor retention of patients starting on opioid use treatment. So the idea to get around this precipitated withdrawal came out of this 2020 paper recommending this approach known as microdosing. So instead of giving someone a therapeutic dose of buprenorphine to start them off, where it will bind or in large scale bind the mu opioid receptors, Instead, if you give them small doses as the fentanyl that they were on begins to decrease in the body, you begin to slowly sort of exchange the receptors binding for a full opioid agonist with that of a partial opioid agonist. So that slow exchange can be achieved through this slow dosing of buprenorphine. So if you look at this dosing protocol, it's 0.5 milligrams sublingually every 3 hours, then 1 milligram sublingually every 3 hours over 2 days. So that means you have to give 8 doses one day, 8 doses the next. That's 16 doses in a day. So, I mean, this looks great. You get decreased risk of precipitated withdrawal that has been noted in case series. But the only issue is there's a large working nursing workload. Here, as I said, it's occurring over a course of 2 days. The nurse has to be giving the medication every 3 hours. Typically, the patients will have to be institutionalized or in a hospital setting for this to occur. So I was approached by a collaborator, Dr. Puya Azar, who works at BGH, and he said that, you know, there is this other formulation of buprenorphine, these transdermal patches that are used to treat severe persistent pain and cancer. And he asked, what could we do, or how could we dose this to sort of mimic what we're doing in these microdosing situations? A lot of times the patients he were dealing with couldn't keep them in a hospital setting for 2 days. They would self-discharge, and he would miss his chance in order to get them onto opioid use or opioid agonist therapy. So the whole idea here is, can we repurpose this formulation to better meet the needs of these patients on opioid use disorder when initiating them on buprenorphine therapy? So what we did is we took a population pharmacokinetic model, or one of these empiric models that was being developed in the literature, and what we did is we repurposed it or used it to perform dosing simulations to say, we know the pharmacokinetics, or we know what the dose-response relationship looks like on these microdosing protocols. How can we devise a patch dosing protocol to mimic this? So through these models, we were able to mimic different types of patch formulations or patch dosing strategies that mimic sort of what we were seeing in the microdosing scenario. In the end, what we went with is this green line of applying 6 patches to the patient, waiting 24 hours, then applying another 6 patches. So in the end, they would have 12 patches on their body. So this is a really out-of-the-box solution, because the maximum sort of regulatory recommended dose of this is 1 patch. So in terms of what this looks like on a patient, something like this, it's almost like a turtle shell, I always think of it. So this is what it looks like when it's being tried in a case series at VGH. And what we've found so far, and what we've published, is that it has worked in these small sort of case series scenarios. And right now at VGH, we're trying to take a look at a larger swath of patients to sort of further create some evidence for this approach. And the great thing is, this approach, besides it looking quite heinous on the picture, these patients can leave hospital, come back after 24 hours, get their next set of patches, so they can be mobile, whether they're in a SRO or in these different housing situations, but still allowing them to get some type of viable opioid agonist therapy. So really bringing in these populations that may not have been able to be treated in an in-house or hospitalized setting. Okay, so in summary, or for my piece, pharmacometrics has the power to inform dosing, particularly for opioid agonist therapy, and improve the care of patients with opioid use disorder. And model-based drug development can be incorporated into several stages of drug development, either the early preclinical, first in human, as well as the clinical stages. So next, Sahithi will talk to you a little bit about her research. You want to switch computers?
Yes.
You'd have to go on the Zoom first.
I have this one.
Okay, perfect. Okay. Sorry for the technical things for everyone online.
Okay, let me do this.
Sorry. You need. Okay. And just put your mic on, and I will.
just need to swap this. Slideshow, that should work. Yeah, I got it. Okay, that should be good. I hope everybody can hear me on Zoom and in person as well. Okay. So there's no doubt fentanyl is causing this, as Dr. Maharaj mentioned, fentanyl is causing this unprecedented burden on about the opioid overdose crisis. This is basically a very highly potent synthetic opioid that is used to treat cancer patients. And because of this, because of fentanyl, there is an increased. Just a second, I'm just trying to.
I think you hit that thing.
Yeah. Okay. So yeah, as you could see in this very province, BC, we have an increased percentage of fentanyl deaths over the last decade. As you could see in there is a huge shift from 2014 to 2016. With this increase in the percentage of deaths in fentanyl, I'm going to go into like what we did to kind of help with this. So in 2016, due to a twofold increase in the percentage of opioid deaths related to fentanyl, the provincial government of BC announced a public health emergency, and a lot of help has been deployed since then. However, at this point, when researchers looked at what is the fentanyl usage patterns in this population, it was mostly unintentional. People were not knowing that they were using fentanyl, but because of its wide presence in the unregulated drug supply, people are being exposed to that. That's what is causing the fatalities and the overdose and the toxicity. However, to help with that, the province came up with several harm reduction services. One of the major important harm reduction service that was rolled out at this point in time is the drug checking services. So the whole idea with this is that people are going to come in, test their drugs, and know that fentanyl is present in the sample that they're using, and they'll stop using it. Is what we were expecting as the idea behind rolling out this drug checking services, to educate people on what is there and how bad it could be to them. However, to our surprise, when researchers kind of studied in the following year, in 2018, when researchers kind of studied, what are people using harm reduction services like drug checking services are using? They found that 40% of the people are actually intentionally using fentanyl at this point. This came out of nowhere as a huge surprise for us, because the whole idea behind all this is to prevent fentanyl fatalities, but people are going and using it very intentionally at this point. Over the last 5 years, between 2019 and 2024, the percentage of fentanyl deaths remained consistent in the last 5 years, approximately around 80%. That is a huge burden. However, in this point of time, we have noticed the substance use patterns, especially all this data is from Vancouver, BC. We have noticed that people started having a huge preference for fentanyl, and various treatment strategies were kind of developed in the last 5 years to kind of help treat people using unregulated fentanyl, to basically help them stop using unregulated fentanyl and obviously decrease the mortality associated with this. There are several treatment strategies. Dr. Maharaj mentioned the first one, that is the OAT therapy. That is basically you kind of give them opioid-like medications to kind of replace the unregulated fentanyl with more controlled and well-studied medications like methadone, long-acting morphine, or buprenorphine. However, with this approach, we have seen terrible dropout rates. People would drop out of the treatment itself, potentially because of two major reasons. They needed a very high doses of this buprenorphine or methadone to kind of match those fentanyl effects. But however, the protocols that we were using followed a very slow titration. So it took us like weeks or months to get them at a very comfortable place. That kind of made them to go into withdrawal, and that's why people started dropping out of these treatment protocols. A more realistic approach has also been implemented right after knowing that there are a lot of treatment dropout rates, is the low potency opioid. So people, what the physicians started doing is that they started prescribing low potency opioids, that is either heroin, that is diacetyl morphine, or hydromorphone. However, at this point, people were exposed to fentanyl on a daily basis. So for them, if you wanted to switch them from somebody who is exposed to a high potency opioid on a daily basis to a low potency opioid, that often led to very low efficacy, and people are also not retaining this therapy well. In the last couple of years, various safe supply of fentanyl programs have been piloted. The two very well-known programs are the fentanyl patch programs, where people were prescribed fentanyl patches, and the other program is the fentanyl powder program, where a powder of fentanyl is made and is being dispensed in a safe injection facility where it's a directly observed therapy. They'll have to use it in the facility itself. However, with this, again, we do not know what the start dose is. Like, where would you start a patient when you're transferring them to unregulated fentanyl to a patch or to a powder program? Again, it requires a dose titration by prescribing them PRN, that is, like, use as much as they need, and finally understanding what dose to put them on. Again, dose is a very important parameter that you would want to know. However, there are two major knowledge gaps. So if you're envisioning a safe supply program that could be rolled out to a much higher population, there are two major knowledge gaps. The first knowledge gap, which clearly surprises us, is that we know that people are using fentanyl for over a decade now, but we do not know how much they're using at this time. That is kind of really sad. And then other knowledge gap is that we don't know what effect the concomitant medications would have on the fentanyl exposure that you're going to prescribe, or that they're using that you're using from the unregulated supply as well. So part of my thesis kind of addresses these two knowledge gaps. The first knowledge gap, the part of the project that addresses the first knowledge gap is kind of quantifying the daily amount of unregulated fentanyl use in this population. For this, we collaborated with Dr. Lean Ping T. Dr. Lean Ping T runs the drug checking services at the British Columbia Center for Substance Use. So what is drug checking services? So what they basically do is that you can go to them with an unregulated drug supply sample, and they'll test that sample they use two approaches, basically. One is a quantitative approach and one is a qualitative approach. The quantitative approach is by FTIR. So if you give them a sample, they kind of scan the sample. It is non-destructive. So they kind of scan the sample and they tell you the percentage of substances that are in there. It is not only fentanyl, but they also detect all the other substances that are out there as well. And the other approach they use is a qualitative approach. It's just an immunotest results. What it basically tells us is it positive for fentanyl or the benzodiazepine or any drug of our interest. So what they basically do is that they publish the results on the website. This is the website. This is what it looks like. And they also roll out alerts for the substances that have very high concentrations of fentanyl or very high concentrations of xylazine or any potential harmful thing to the public. When we first met Dr. T, what they were doing is that they were also doing this survey to kind of assess the effectiveness of the drug checking services in the province. And they were collecting various patient-level factors information on how are they administering fentanyl, frequency, route, location, and along with various socio-demographic factors as well. So for our purpose, based on that, I have some results from like how frequently fentanyl is being used in this population. So we looked at how frequently people are using fentanyl. This data is from the past 1.5 years or so. We had a sample size of around 140 individuals, and 90% of them said that they're using it on a very daily basis. And we also asked them, like, how would they use it? Most of it is inhalational, 71%, and only like 27% are injecting it at this point. Moving forward, so the whole idea is to not understand like how many times they're using, how often they're using, or what's the route of administration. The whole idea of the project that I'm focused in, like, this information is already there in the literature. Like, it is very well known at this point. But the whole idea of my project is to understand how much amount of fentanyl they're using. So we had around 143 samples. Sorry, the N is missing from there. We have around 143 samples. So what we added is that to the existing drug checking questionnaire, we added 2 questions. One is what is the typical amount of the substances with fentanyl that you use? And the other one is the lowest and the highest amount of substance they're using. We asked these questions, we had the response, and then we integrated with the FTIR results. So they're saying how much amount of the substance that is in their hand that they're using. And we are also testing the same substance to kind of determine how much amount of fentanyl is actually present in that sample. So we had a percentage of fentanyl present in the sample from the results, and we also know how much they're using. So we plugged both the information together. For example, here, when we did the interview, let's say that they're saying that they use 2 points of fentanyl. So we know that a point is 1/10 of a gram. If you ask us how we know it, we just interacted with people using substances and we asked them, what is it? And they told us that it's 1/10 of a gram. It's 2 points of fentanyl in a day, you convert it into grams. And then let's say the FTIR results showed that there is 5 to 10% of fentanyl present in the sample. The proportion of fentanyl would be 0.05 and 0.10. So you'll have an estimated range of like how much amount of dose are these people exposed to at this given point. So it can be anywhere between like a 0.1 gram to 0.02 grams. So that is around like 10 milligrams to 2 milligrams. So that's not that low, but 10 to 20 milligrams. So that's not that low, however. So once we calculated these values, like I said, I asked them how much they would use on any typical day. I also asked them what is the least and the highest amount that they have used so far. So when I plugged all this information together, this is how the data was looking like. The x-axis is the dose, that is in milligrams, which kind of seems a lot because it's a very, very positively skewed data, like as you could see here. And this is just basically a density plot. The green one is the maximum dose. The blue is the typical amount that they've reported. And the red one is the minimum amount of dose that they have reported. So I basically, since it is a positively skewed data, my mean estimates would be like very, very, very badly influenced by the highest amounts. So I took out the median estimate of what is the minimum, maximum, and the typical median estimate looking like. So I had around 87.5 milligrams of fentanyl as a typical amount that this patient population is using, 37.5 milligrams, and the maximum amount, 192. If anybody is a clinician out there, you would be straight out thinking I'm crazy or this whole study is flawed. I agree with you because the highest dose of fentanyl patch that is out there is 100 micrograms per hour. That gives you around like 2.5 milligrams per day. So if I'm saying that they're using like approximately 90 milligrams and the highest amount of stuff that is out there is like 2.5 milligrams per day, you definitely think I'm crazy, but we have a way to validate these results. Luckily, we have some collaborators at VGH, Dr. Puyazar and his group. They were doing this symptom-inhibited fentanyl induction protocol to kind of, they're doing this protocol to kind of initiate opioid agonist therapy, that is like equipotent dose of how much amount of fentanyl they're using, and also meet the opioid needs of like just the hospitalized patient itself. They used to have, they do consults for people using unregulated substances. If they're admitted for a different surgery or a life-saving procedure, they'll have to give them some amount of opioid to keep them in the hospital without withdrawal. So they were testing out this protocol to kind of meet the needs of those patients. So what they were basically doing is that they were giving either 400 or 800 micrograms of fentanyl every 5 minutes until the patient is comfortable in the hospital. When they did that, they did that and they published a very case series very recently. And for one of those case series patients, they have given 9,000 micrograms of fentanyl at a single instance. So and then what they do is that to understand the daily fentanyl dose requirements in these patients, they kind of multiply it by factor 8. And then that leaded them to like 72 milligrams of fentanyl in a day. Compare this with what we have estimated. It is not perfect, but it is in the same ballpark. And the published case series had like 2 patients. And we had like 140 patients looking at it. And most of the population that we studied are also inhaling the drug. So a part of it might also not reach the system in that case. So when we look at that, so if you're wondering where the magic number 8 came from, so before they multiplied it with, before they started multiplying it with 8, they gave patients fentanyl for 24 hours in the hospital. They admitted the patients into the hospital. They gave fentanyl to the patients whenever they needed it for 24 hours. They studied a bunch of patients about like how many times they needed it in a day and how much they needed it. That's where the 8 came from. They needed it 8 times in a day. And it kind of matches with the PK profile of the drug because the half-life of fentanyl is around 3 to 4 hours. So if you want to maintain the same amount of the drug that you have given in the beginning, you'll have to dose 6 to 8 times a day. That kind of adds up here as well. So based on this, we are confident that whatever we estimated is around the ballpark. And this is the first study to quantify the daily dose of fentanyl that people are using. This is it seems like a very small project, but it has a huge application potential. One, it changes everything that we know about right now. Like we don't even know where the dose is. So it changes everything about the protocols that are there up right now. So one, it is very applicable in the sense that the starting dose of fentanyl safe supply programs will be drastically higher than what you would do without the study here. And the other thing is that it'll give this idea to the researchers that, oh, they need 87 milligrams of fentanyl or somewhere around that ballpark. And you cannot give them like 20 milligrams of methadone and hope that they'll do okay on that. So it'll help us develop these new oat titration protocols that can mitigate the risk of withdrawal that is happening due to insufficient dosing. Develop appropriate opioid dosing protocols for hospitalized patients, very, very crucial so that they don't just leave from the hospital without getting the life-saving care that they would need. Okay, as I said, the first project, the first project is focused on the first knowledge gap, and the second knowledge gap is to understand how this fentanyl exposure is kind of influenced by co-administered medications. Dr. Maharaj luckily gave some intron PK models, and I have a very small section here. The physiology-based, I'm planning to do it using the physiologically based pharmacokinetics models. These are the complex models. However, why is this important? If you ask me, people using unregulated fentanyl often need treatment for other stuff like HIV, because HIV is very prevalent in this population. Medications like cobicistat or ritonavir, these are the HIV medications. They're used to treat HIV and can interact with unregulated fentanyl. How do they do that is that fentanyl is basically a substrate of a liver enzyme system called CYP3A4. And the CYP3A4 metabolizes fentanyl into a not active metabolite, that is the norfentanyl. And this norfentanyl is basically eliminated in urine very quickly from the body, and it has no like toxic effects or anything that sorts. However, if you have ART that has cobicistat and ritonavir, these medications tend to block the CYP3A4 enzyme. So what happens is that in turn, the conversion of fentanyl into its metabolism is inhibited. As a result, what happens is that the fentanyl will start building up in the body, and it can increase this cobicistat and ritonavir can increase the exposure of fentanyl, leading to the adverse drug reaction and toxicity associated with fentanyl exposure. So typically any drug-drug interaction information. So what do you do if we are starting HIV meds in patients using unregulated fentanyl? So oftentimes we rely on clinical studies for such information about like how much exposure change would happen if we were going to start with them on ritonavir or cobicistat. But however, in our case, the information in the literature is limited. Like I have one study that is looking at ritonavir-based interaction, but I do not have any information on how cobicistat, how this interaction would look like with cobicistat. So when such information from clinical studies is not like available, one other approach to address this knowledge gap is to use PBPK models. These are basically computational models, computational pharmacokinetic models that can be used. Okay, what are PBPK models? These are basically in silico math models. Like they're very similar to human body, in the sense that they have multiple compartments, like we have multiple organ systems, and each organ system's connection to the venous and the arterial blood supply is depicted by the arrows there. that determines the direction of the blood flow as well, which is also very physiologically relevant. And what these models use is that they use a series of mathematical equations to determine how a drug moves from one compartment to the other and how the drug moves from the blood supply into the tissue as well. And one very cool thing about these models is that they're approved by the regulatory authorities to predict drug-drug interaction. So how do we make these models? The whole objective of this part of my thesis is to develop a fentanyl, a PPK model of fentanyl to predict fentanyl corbicostat drug-drug interactions. How do we make it? Is that you have two major parameters that are going in there. Actually three, but the third one can be optimized. The two major things that are going are the physicochemical properties that kind of talks about mostly the chemistry of the compound, like how lipophilic is the compound, what is its pKa, what's the solubility of the compound in that sense. And the system-specific parameters are mostly the organ-related parameters. What's the weight of the human? What is the weight of the liver? How much blood is there? What's the weight of other compartments in the body, kind of. But luckily, I didn't have to go deep down into figuring out the weights and stuff. This is already there in the software that I use. It is an inbuilt system that is validated. So this is all there, and I just have to go into the literature and find the properties of the drug that I'm interested in and combine it with the PK information. This is also very important if you ask me. Like there are some parameters like fraction unbound of the drug in the blood. Information is what you need here. It's not only very specific to fentanyl, but you can, to build any PPK model, this is how you go about it. Like you go into the literature, get this information, understand its PK, understand the mechanistic PK, get that information from the in vitro studies, add it into the model, and then you have a basic working model. It needs a lot of optimizations to make it agreeable to the observed data. However, here, this is a workflow of the project that I'm doing. So I'm interested in predicting fentanyl and corbicostat interactions. But however, if I'm predicting a brand new interaction, like Dr. Maharaj mentioned earlier, the regulatory mandate is that I need to prove that my models are working with a studied scenario. Luckily, I have that study scenario, that is the ritonavir. I have data on ritonavir, on how fentanyl interacted with ritonavir. So if I say that my model is performing well with ritonavir, I can predict it with corbicostat. So for this, I'm going to build model of, I started building models for ritonavir, fentanyl, and corbicostat separately. I validate it with the observed data that is out there, observed clinical concentration time profiles that are out there. And I predict the fentanyl ritonavir, I simulate the fentanyl ritonavir drug-drug interactions, compare it with the observed data that is already existing in the literature, and say that, yeah, my model is doing good. And then I move on to predicting fentanyl and corbicostat drug-drug interaction. I have some preliminary results from this thing. So like I said, let me orient you to the graphs. The x-axis is the time, and the y-axis is the concentration of fentanyl. These are the results for the fentanyl model. The lines are the predicted concentrations from the model. The dots are the observed concentrations from the clinical studies. The dots there are also, the dots on the right are also the observed concentrations, but the error bars are the error in the concentrations that is seen clinically. In an ideal world, I want my model to pass through all the points that are right there, but I think we can get there, but needs a little bit more work. But so far, it's doing reasonably okay. And then I was excited to see, like, can this predict the ritonavir drug-drug interaction or not? And then I simulated that out as well. So the one in blue is basically the control scenario where fentanyl is administered alone, and the one in the orange is the test scenario where fentanyl is administered with ritonavir. It looks like the model is doing fairly okay. However, we would want to improve these predictions right there. So it needs a little bit more work from here. And then once I have this, I'll just have to I'll have to develop a corbicostat model to be able to predict. The next step for this is it needs a little bit more optimization, like I was mentioning with the fentanyl predictions. I think one way of addressing this, a small difference in the predicted and observed is to include the metabolite model also with the parent model. That could kind of tease out some of the parameters that we are looking at. And another important piece would be to develop this kubicostat model and predict that interaction and like kind of understand how is it looking like. And then other important piece that I was interested in is that all the clinically done drug-drug interaction studies up until now are based on the standard dosing of fentanyl. But if you look at the standard dosing, the 2.5 and the 19 milligrams that we're looking, they're very different. So we were just curious about predicting, using this developed framework to predicting the drug-drug interactions very specific to people using fentanyl with the doses that they're using as well, to kind of see everything would be what it is looking like in the literature or not. That would be the interesting application piece for this model. And we can take questions with that. Yeah. Yep.
So I did a study that was last about dosing, and it was a bit difficult to collect that data because we had individuals saying they consume a ball, a vegetable, or a paper, or street terms, and I was wondering how you were able to figure out how much dosing that would be. We had to outsource for the personal lived experience, but yeah.
Yeah, so what we did is basically is that the drug checking services also have a technician that work there, and the collaborators that we have been working in this for quite a bit, and they know that street conversions of few things. Like I know I even went down there and I asked them how much is a point. They all had the same answer. It's 1/10 of a gram. The amount of fentanyl that they got in hands would be different, but with the money involved and with the dealers involved, we wouldn't expect huge differences in terms of like how much they're being handed out, because it's all like the street supply, right? But we did have information from patients who are using fentanyl saying how much a paper and a ball is as well. So when we asked the question, we did ask, I think the interviewers asked them like how much is a ball, and then that kind of matched with whatever that we are using as a standardized conversion sheet, and then that's how we did the conversion for the grams. Did that answer your question?
How much is a ball?
I think it's 8 ounces. It is 8.
8 ounces.
I think it's 8 ounce. I don't remember exactly on top of my head, but I know I'm very familiar with the points, but they did have a conversion that we were using. Yeah. I think give me one more second. I have that. Yeah. Yeah, I think I have a similar thing with me. Just a second. Yeah, I think it's about right. Sorry, I must have been mistaken in that sense. Yeah, this is what we have from the study itself. $20 is basically 1 point that you basically get. 1 ball is 3.5 grams, like you said, half a ball, it's half of it, and 0.05 is a paper. Ounce is 28 grams, which was very rare that we had such high numbers.
So it's not very high average dosage. The sample says we ended up with 120 doses. It was 1.68 grams average.
Yeah.
In day. And I think yours was, you said it was 0.2.
The grams? Yeah. So what I think we are looking at 2 different estimates here. So
like, or is it in a day?
It's in a day, but what we did is that if somebody is saying that they're using a point of fentanyl, right? The very typical concentration of fentanyl that is in a point is around like 10% of fentanyl. So even though they said that they're using 100 micrograms of fentanyl, they're not actually using 100 micrograms of 100 milligrams of fentanyl. They're using 10% of it. That is 10 milligrams.
Is that it's high because of how
It is I think your estimates are so high because that is the main problem why this has never been quantified before as well, because we do not know, like we know the street quantity of how much they're using, but we do not know how much amount of fentanyl is being present in that. So with this project, what we tried is that we wanted to also analyze how much amount of fentanyl in it so that we can give an accurate response rather than giving a very huge estimates. If I just look at whatever they say, it's going to be very huge. But when you integrate it with the drug checking services or analyzing the sample, that's when you get the real estimate. But these grams, I was actually thinking about it, about like how true can be these weights, right? Like when they say a point or when they say half a gram or something, how true can this be? And then I've also thinking like it could be true because one, they're paying for it, and the street supply is very brutal in that sense. The money talks, right? Like if you don't get enough, there's always fights and violence. So I think since the money piece is involved in this whole thing, we were hoping that these estimates would be correct. But again, this study has this bias of recall bias of you don't know how much they're saying is absolutely right or not.
I'm not sure if you mentioned already, but do you find that the price changed over time, like the price and the amount, basically like inflation, but with
I think it is true for everything we eat, like bread and butter, but not for this. I started my PhD 3 years ago. You get a point for $20 then, you get a point for $20 now. Yeah. This is very well established as well. It is from lived experience. And I personally checked some of these values by asking them, but I just didn't remember it on top of my head. Yeah. Yes. Yeah, that is one of the interesting piece that we wanted to do in the second piece of analysis, like kind of relating it to not only just that is also relating it to like different patient level factors. We had access to this huge data set that kind of collected data on like homelessness, location, where they're located in BC. Is it in Vancouver? Is it in Kelowna? Is it in Abbotsford or things like that? And we also have information about route, frequency, and what are the drugs they're using. So we wanted to explore this idea of performing a generalized linear model to kind of figure out if other factors are affecting how much they're using. So what are the risk factors for people using higher doses of fentanyl is what we wanted to identify as a second part for this work as well. Yeah.
To try to help to understand the relationship between like fentanyl or opioid exposure, other drugs, and mortality from overdoses. Like when there's a fatality, they do blood work to determine what was the cause of it. Is that common or?
Yes. So what the coroner does, the BC coroners, who assess the deaths in like any scenario, they test out every single drug that is that they take the samples and they test drugs. That's where you get those graphs from. Let's say like the percentage of deaths with fentanyl is those deaths, the samples from the people who passed away have tested positive for fentanyl.
Model fentanyl exposure, whether or not other drugs were present, like to determine to come up with a model of mortality risk or.
So right now what they were doing is that they had a cutoff value. Like it seems very like empiric, but they had a cutoff value for 50 nanograms per mil of fentanyl. So anything over 50 nanograms per mil is considered a huge exposure of fentanyl. That is considered as a very huge exposure of fentanyl, and that has caused the death is what they're doing right now. But again, dose is a very critical thing that you would want to know in these patients as well, right? Like you want to know what dose caused the death. Like that's if you want me to put it right there. But however, it is quite hard because you're dealing with people who are not alive anymore. They cannot say how long ago they took the drug, what their tolerance was, or how long they've been using it. So that is pretty tricky. Yes, it is very hard to model with the missing key variables like the dose and with with samples from cadavers, it is known that a lot of redistribution also occurs. Like fentanyl is highly lipophilic, so it tends to be in the tissues. But it is also studied in the literature that once a person passes away, the drug redistributes to the blood. So, and that's also tricky to kind of understand because we don't know the whole mechanistic side of things.
Maybe understanding like non-fatal overdose.
Yes, that's what they're trying to do with the arbitrary cutoff, but it's very hard to like say with that cutoff as well, the 50 nanograms per mil that they were using right now.
That's very arbitrary.
Yeah.
Our data set has self-reported non-fatal overdose by the participants,
Yes.
but it's not linked to the mortality data.
Because that is at this point, right? So you're looking at like previous overdose history. But one other thing that we propose is that now we know the doses that they're using, kind of following them up to the future. And if you have access to their PHN adding up, then you could kind of understand what is causing the mortality. But right now you cannot go that way. You can go only towards the other side of data collection, that is past experiences with overdose.
It was kind of consistent from 2016.
Yes.
Overdose that. However, that didn't shift anything because it kind of changed from illicit drug.
There is
has changed.
Yes.
Or taking more fentanyl and yes, unbeknownst.
Yes, but you see what I'm presenting here is basically the percentage of fentanyl deaths. It's not the actual amount of deaths. The deaths there is a dip in the 2019 data that if you see from Dr. Maharaj presented that raw data set, where there's a dip during the pandemic because we couldn't assess the number of deaths during that period very rigorously. But what I presented is mine is a percentage of deaths with fentanyl that I've showed. So that was consistent across. It's not the actual amount of deaths.
Bigger percentage of death from fentanyl during the COVID.
We haven't seen, this is just the data itself, right? Like we haven't seen anything as like the increase in the number of deaths from fentanyl itself. It is pretty consistent with like somewhere around 83 to 87. It is
when I look at 2019 to
there is
an increase.
Yeah.
Sorry, it's hard to see, but this one's 2019 and then 2020. This is the raw rate of unregulated.
What was there a dip in 2019?
It's the time of the COVID year. So we 2019 towards it's called 2020, yeah.
2019 shouldn't technically, because
But-
it started around the end of the pandemic. Because
Yeah, but there is an explanation for it in the BC Corners website.
2019.
Yeah, the 2019 data, they mentioned something about the pandemic and not being able to.
Unless they're assessing the deaths post, like they take the samples and they don't test it right away. And
That's what they do. Actually, the percentage of fentanyl deaths is also like, that's what they do, right? Like if a person passed away right now, you do not know it is caused because of fentanyl, but post at some point they analyze those samples and then add it that this death is due to fentanyl. I don't know if that made sense.
Well, push it further away. If they took the samples in the year of COVID, say 2020,
Hmm.
take some time to analyze them and then they
Yeah.
post those figures, it would push that number ahead in time.
No, I think that what they do is that those 83, 84% will change with time because the more data is coming in, right, about the death that happened. Let's say that a death happened today and then they collected a sample today. The death is marked on today, but if the fentanyl causes death or not is added sometime like 2 or 3 months away from it. That's when they know that fentanyl is detected in this person's sample. So that's when this number changes.
Doesn't explain the dip.
On the 2019, yeah. I think I remember seeing something on the BC Corners stuff, the Corners website.
Yeah.
Reports, like they'll end like October or something, and then the next year, like is from like November of the previous year. I don't know if that was the case here, but sometimes that does happen.
Yeah, I think it's just a raw data set from the BC Corners website. There is a potential explanation, but I cannot remember on top of my head. Yeah. Yeah.
I think they were actually more successful in reducing the use. I'm
Yeah.
going back to your slide about Paracelsus and toxism.
Oh.
And did he really say that everything is toxic? Or is he just trying to do it with respect to
There's
molecules that are used in clinical practice or whatever?
So I was around in the 1500s. I don't know if he was talking. Like I think he was a physician, so I'm assuming he's talking about some type of medications or.
Yeah.
Well,
So
probably
everything
didn't use
has a
that.
therapeutic index, right? It can put
So it's
any
the
drug
word
in.
everything, so
Yeah,
it actually says everything. But so it's translational error.
And I assume that was Swiss, so. Eventually was probably very difficult. There's so many words that maybe don't.
Yeah.
Okay, I don't see any questions on Zoom, so thanks again, Anil.
Well,